正的为儿童的发展而研究儿童，而不是用儿童语言做材料去研究别的理论。当然，这一条路也可以走，也可以走。所以我今天主要的目标就是希望儿童语言研究真正能够帮助到儿童，帮助到家长，帮助到教师，帮助到儿科医生帮助到所有给儿童相关的这些人群。这是我今天报告的一个目标，也是我最近思考的一个问题。我把它叫做为儿童的儿童语言学研究。下面讲第二个问题：儿童语言发展这么奇妙，那么儿童语言系统和百科智慧系统究竟是一个什么样的关系？这就是我们经常讲的语言和思维的关系，语言和认知的关系。那么实际上，大量的这个事实都在证明，语言不是一个独立的系统。语言这个系统是添加在百科智慧系统之上的，它是百科智慧系统的明珠，也是与百科系统协同互动发展起来。所以，语言和认知、语言和思维不是一种相互对立、相互孤立的关系，而是一个相互依存的关系。那么就百科系统而言，现在大量的研究证明，它是由3个子系统构成。一个子系统，它是知识化了的系统，也就是说，由于人类科学的发展，对于世界已经知识化，但是人类的百科系统还有更大一部分是没有知识化的系统，是常识系统，是感觉系统。除了知识化的百科知识和未知百科化的未知识化的百科知识，人脑的各种认知决策机制，也是百科系统一个最重要的。那么语言系统，它实际上是不断地扩充住百科系统。由于语言存在，那么语言帮助我们发展认知，来扩充住我们百科智慧系统，逐渐地把那些未知识化了的知识化，把那些未进入我们百科系统的进入到我们百科系统。同时，百科智慧系统又支持住语言系统的运行。没有百科系统，只有语言系统是没法运行的。那么这种情况，我们把它叫做语言百科孪生，它是一个相互感知的这种孪生关系。所以，这个语言百科孪生是我们提出的一个假说。那么除了语言系统和百科系统之外，我们人类如果我们把语言系统和百科系统看作是我们的灵，那它还有一个肉的系统在支持，支撑住语言系统和百科系统的是我们物质的人脑和身体。人脑和身体我们可以叫做脑身系统。那么语言系统、百科智慧系统和脑身系统的关系像右图所示。那么灵肉之间也是一种协同关系。有研究发现，帕金森病患者都有运动问题。那么这些帕金森症患者，他们在处理动词的时候也会出现相应的问题。那么因为我们身体各个部位对应的脑区 和语言区域是相辅相成，是相辅相成。那么姜孟教授啊，他曾经有一篇文章，就是讲帕金森症患者这个动作动词在加工的时候啊，和他的这个身体障碍基本一致。当他这个跳动有困难的时候，他在处理跳的语义的时候也有困难。那么这些研究不仅在汉语里面，主要是通过英语，通过其他这个语言得到了证明。那么一种重要的解释就是运动和语言相关，运动和语言相关。那么这个帕金森症患者、老年痴呆症患者还有很多语言疾病患者的情况，说明语言和运动和脑的部位都相关。因此，我们可以进一步提出灵肉协同假说。不仅是语言和百科是孪生关系，语言、百科系统和人类的脑系统，甚至和身体的系统都应该相关。那么，人类的语言活动不仅有百科系统的支持，而且还有语言系统和身体系统的支持。那么，人类的脑神运动，回过头来，我们可以说都有语言系统和百科系统的支持。在人进行运动的时候，人类的百科系统和语言系统也在运动，也在支持。当人类进行语言运动的时候，我们的百科系统、我们的脑系统、我们的身体系统也在支持。那么，语言障碍者的身体运动为什么和我们正常人不同？就是因为他缺乏了大脑应有的支持，因此缺乏了大脑语言的支持。百科系统虽然可能还存在，脑系统、身体系统还存在，但是由于语言障碍问题，所以也会带来我们身体的问题。这就是灵肉协同假说啊。那到现在为止，我们提出了2个假说，一个是语言百科孪生，一个是灵肉协同。那么这种假说对于我们母语教育、外语教育会很有一些启示。就是我们在语言教育的时候，不能只关心语言系统，还应该重视百科系统，甚至重视脑神系统的体验。我们为什么说语言学习需要体验，知识学习需要体验，也就是需要脑神的支持。没有脑神的支持，也就不可能有生活的支持。离开生活的支持，语言学习和知识学习都将是空洞的。那么一些语言病例的康复，以及老年语言能力的维持，不只是语言系统的康复与锻炼，还要注意3个系统的互动，包括老年这个语言、大脑、身体之间的互动都非常重要。那么，计算机语言智能的获取，现在主要是数据驱动，实际上是非常偏颇的，已经遇到了天花板。为什么？因为只有语言数据的学习是远远不够的。计算机目前最难的是关于没有知识化的那一部分百科智慧，而且缺乏人脑的这种决策机制。我们人脑的决策机制最重要的就是小数据决策，这个和计算机恰巧不一样。计算机需要大数据决策，人脑需要小数据决策。极端化的状态，零数据都可以决策。这个我们很多女同志经常没有任何蛛丝马迹就能发现她的先生的这个活动的轨迹，都是零数据决策。 我们很多重要的军事活动、军事决策，在对方这个信息非常不透明的情况下，能够一些军事家能够做出很好的军事决策，这是计算机不可能做到的。所以，计算机的语言处理、智慧处理，目前仅仅用大数据，满足于大数据，不可能获取真正的人工智能。那么，既然儿童语言发展，它既有先天的语言习得机制，也有后天的语言输入的影响。那么，人脑的先天机制在短期内你是无法改变，它是受遗传因素制约的。所以，真正要改善儿童的语言发展，还是要看它受后天的影响，这个是你可以控制的，可以改变的。从理论上来说，我们认为儿童语言发展是先天的语言机制和后天的语言接触相互作用的结果。但是就人类来说，真正要改善儿童的语言发展，先天的语言机制，你很难在短时期内改变，那是要靠长期的遗传因素、基因活动才能够改变的。所以我们能做的，那就是研究儿童受到后天的哪些语言的影响。这是一个策略性的举动，也是对儿童语言发展他的认识。因此，这就引导着我们要去研究，究竟谁给儿童的语言发展项目，我们把它叫做涉耳人群。这就是我要讲的第3个问题它的逻辑上就是先天的问题，短时间很难改变。我们要帮助儿童，那就要研究谁影响儿童，影响儿童的语言发展。那么现在看起来，语言输入包括广义的儿童语言环境，对儿童的语言发展是具有重要作用。我们看待语言不能只看语言的符号系统，还要看儿童的语用发展。如果说儿童的符号系统发展先天的因素占很大作用的话，那么儿童语用的发展，就是怎么样能够把话说好说得体的发展，更重要的是后天的影响因为我们看到儿童在发展当中，一开始小的时候说话不得体，逐渐的学会在什么样的环境下对什么样的人说什么样的话，这是一种发展技能。这个技能不是每个人都能够学会的。如果是先天决定的话，每个人的说话水平应该是一样的。事实上，人与人的语用能力差别是很大的，说明受后天的影响。那么受哪些人的影响是我们值得研究的？现在看起来应该从4类人群里面去看第一是家庭，第2是学校，第3医疗单位，第4社会其他行业。对儿童语言发展影响最大的，无疑首先是家庭。那么在家庭当中，对儿童影响最主要的是父母。我们说，父母与儿童交谈是儿童最好的教科书。儿童的语言就是父母的镜像。有人经常讲，我怎么教我孩子学说话？正确的回答是，你怎么对孩子说话，孩子就怎么对人说话。当前我们家庭最大的问题是父亲缺位，这是我们每个家庭面临的最大问题。 包括到幼儿园、到小学，都面临着男性缺位的教育。因此，从儿童语言发展和心智发展上，我们必须希望父亲要多陪孩子。这个幼儿园、小学要多有男性教师出现，不然的话，这个孩子的语言输入啊，它就是不全面的。汉语还好，这个性的这个差别不大。很多语言里面，这个男性和女性说话是不一样的啊。这个男性缺乏呀，经常使孩子不能很好的习得这个阳性的很多这个性数格呀这些东西。那么，在中国，在东方和西方不同的是，我们在育儿上祖辈的深度介入啊。那么每一个家庭，在城市，我们都年轻人希望自己的父母这个来帮助照顾孩子。在农村，很多年轻的父母都出去打工了，孩子主要留给了父母。所以，在中国最重要的一个特点，也是东方一个主要特点，就是祖辈的深度介入。这个祖辈对儿童的影响，第一个，祖辈的语言都比年轻的父母的语言比较老，它保存住这个过去的词汇和语言表示的一些情况。而且老年人讲话呀，冒险的东西少，人到年龄大了，干什么事情讲究沉稳。所以在孩子的勇敢性的培养上是缺乏的，而父母倒是对孩子虽然很爱，但是经常鼓励孩子去克服困难。祖辈和父辈对孩子语言发展的影响和心智发展影响是不一样的。而且大量的研究证明，儿童语言学习并不仅仅是你给儿童交谈了，儿童就学习了语言。儿童经常是以旁观者的身份学习语言，包括电视里面放的这个家庭成人交谈的，都是他学习的对象啊。有时候你说我没有告诉孩子这些事情，他都会了，都知道了。这个大家庭啊，我们现在所谓的大家庭就是三代同堂啊，他家庭语言格局是不一样的。那么儿童只有父母亲两代的话，他只知道这个他和父母怎么谈话。那么一旦有祖辈在这个家里的时候，他就知道父母和祖辈怎么谈话的时候，他就学会了这个不同的人与人的交际，这样对孩子也是有帮助的。目前呢，这个很多新的研究就研究祖辈对儿童的语言影响啊，这个最近有些文章还是很新的。第三个就是同胞在儿童语言发展中的地位。中国过去是独生子女啊，没有同胞，同胞剥夺了之后，我们在下面在座的很多可能都是独生子女。那么独生子女的语言特点，在你们身上实际上还是可以研究的。研究你们的语言状况，也就是研究独生子女的语言状况。那很显然，独生子女他由于经常跟成人接触，他的语言发展很快，而且呢成人化很强。但是这个由于成人和儿童经常的很少那种同伴之间的这个交往，同胞之间的交往，这个老大和老二经常最重要的就是他们经常的会相互争、相互吵，甚至相互打。怎么样学会自卫？怎么样学会争论？怎么样学会这个在争论中获取胜利？这是同胞之间交往才能够学。你跟父母亲交往啊，你学不了这些东西。所以同胞剥夺对于儿童很多语用能力的发展是有影响。那过去我们独生子女时代已经有些影响，在我们这一代我们现在的年轻教师身上，是不是看到这个独生子女家庭成长出来他的优势和他某些方面的不足？那么现在呢，我们又开始放开二胎。这个二孩是个非自然性儿，他跟老大的年龄差得很大。 这老大原来是当独生子女在养，这现在突然天上掉下来个林妹妹，啊，很多东西原来一切都属于他，包括父母就是他的，现在反而又多了一个，这样啊，这个像莎士比亚笔下的嫉妒啊，什么东西，可能都会在未来的中国社会重演。这个现在最大的问题是怎么对待老大？对待老二不知道这个情况，但是老大的心理上会有很大的落差，他有时候也开始了解一些世态炎凉，而这个人呢，父母啊，他都是亲那个小的，他什么东西都要老大让住这个小的，老大和老二有矛盾都是批评老大，这样对老大心理上都会产生很大的阴暗。我观察到很多这样的这个双胞胎非自然二胎的这个家庭情况，在父母亲不在的时候，那个老大就会大打出手，在洗澡间里面，在其他地方，出来以后笑哈哈的对父母说：“我对他挺好。”实际上，这个有很多情况在发生。那么对于同胞的问题，我说中国研究儿童语言有3大优势是我们西方朋友所没有的，一个就是祖辈的深度介入，这个是西方没有的。第二个，独生子女，这个时代过了，但是有大量的资料还存在我们的小说里面，存在我们的记录里面。第三个，一个新的机会，一个新的社会实验室，就是非自然二胎，给我们这个儿童语言研究带来了一个这个天然的实验室。除此之外，家政人员的介入这个街坊邻居也都是儿童语言影响儿童语言发展的一些材料，特别是家政人员育儿嫂月嫂这一些，他们如果没有很好的儿童语言知识的话，那么对儿童语言早期的发展影响也是比较大那么这是家庭的4类人群。那么第2类人群就是学校，学校里面学前教师、小学教师、家庭教师，他们和儿童交谈和与儿童提供的知识都是理性的教科书。孩子最听谁的话？教师的话。他认为教师讲的是真理。家长讲的话和教师讲的话之间一比，家长那个话就不是真理了什么都是我老师说的，什么我老师说的。因此这个儿童语言的发展，包括语言知识的教育，学前教师、小学教师起入非常大的。教师怎么对待学生，那对学生早期心理上产生的影响是非常之大。我们还是主张阳性教育这个不要让孩子从小就失去创新能力。我们现在教育的最大问题就是叫孩子听话，扼杀孩子的创造力，这是我们教育家必须关注的问题。往往给孩子一个唯一的正确答案，让孩子接受那些不是他这个年龄段该接受的问题。所谓的正确，实际上并不适应孩子。那么第2类是特教教师。当儿童有了语言方面的问题和心理方面的问题，比如说视障儿童、听障儿童、自闭症儿童还有一些心理有缺陷的儿童，我们往往特教会进入特教学校。这个特教对儿童的影响比一般教师的影响更大。我们到特教那里去，他不仅这些教师要教会儿童特殊的学习工具盲文、手语，更重要的是这些人的心理康复。他们在智慧上、百科系统上，在脑系统上应该没有问题，只是身体系统和这个语言系统、心理系统出了问题。 那么我们怎么去修复它？让这些人将来成为社会的建设者，而不只是成为社会照顾的对象。我觉得这是我们未来人类对待特殊群体很重要的一个方面。所以心智的康复非常之重要。我们不能让这些特殊人群的就业往往是按摩呀、推拿呀这些特殊领域的就业现在往人工智能往网络领域特殊人群就业也是很多的。我们应该让他和我们共享信息、共享知识、共享文明、共享未来。所以从小教育就应该做好。那么在学校里面，第3类人群是校长他们是学校语言规划的制定者。为什么今天的会议我们张校长会来？因为他是学校语言规划的制定者一个学校用什么语言作为教学语言，这个会议用什么语言作为会议语言，都是重要的语言规划。那么第3类是医疗单位。医疗单位在儿童语言发展方面往往被我们忽视，实际上非常重要。首先就是妇科医生。人第一次当妈妈，关于怎么育儿、儿童怎么发展、什么样的儿童指标是正常指标，都是在妇产科那里和妇产科附近得到。我最近的调查才发现，原来妇产科附近有很多社会服务来介绍这个胎教介绍儿童这个发展的很多东西。当然，我作为做儿童语言学的看了以后，我很焦虑，很多介绍是不正确的，是不正确的。那么第2个就是儿科医生我们孩子总是要去看儿科医生的。儿科医生的语言行为构成了我们儿童最小对一的认识这个对一的表达，这个对一的恐惧，特别是对护士的恐惧都是给儿科医生接触造成，因为开始要打针嘛，打疫苗嘛，是吧？那么儿医是人类的天使，是人类3大职业当中这个一类保护我们生命的再一类就是传授知识的教师，再一类就是保护社会公平的司法。所以医对人类太重要了，而我们从小给孩子一个什么样的医的印象，从儿科医生这里开始。而且我们的语言器官、心智器官是不是正常，也由儿科医生来判断由儿科医生来判断。那么在这种情况下，儿科医生他在语言量表方面还有好几个方面比如说他知道这个孩子耳是不是聋了，声音这个量表，他的语言是不是正常词汇量表、语法量表。现在我们医院里面儿科医生用的很不适合中国儿童的量表，很多量表是从国外翻译过来。那么还有就是耳鼻喉科大夫、眼科大夫、神经科大夫等等，他们对于儿童的这个发展的影响，还有这个儿童医用器材的提供商也需要一定的他和儿童有交集，他和儿童要交谈。那么第4个就是社会其他12行业这个儿童读物的作者、出版者、音像制品的作者摄录者、表演者、出版者等等，对儿童影响都很大。总之，这个还有现在给儿童陪伴智能玩伴的时代已经到了，哪个儿童将来都会有个智能玩伴今天我们班教授也在这儿。我们现在给儿童的智能玩伴根本不适应儿童的智慧发展，教个乘法口诀，特别如果随意的联网，网上的很多东西就会进入到现在儿童的生活里目前我让我的一个博士生研究的就是目前中国市场的儿童玩伴里面 所装的这个知识啊、内存啊这些东西，究竟哪些知识这些生产商在供给我们儿童？这是一个很大的课题啊，应该引起我们的关心。所以社会其他12行业也是一个很大的问题。那么刚才我讲的这4类17行人群，17个行业的人群，都是我们儿童语言学应该研究的对象。我们采集的资料不应该仅仅是儿童自己说话的资料，那个是研究不好儿童语言发展的，没有办法给他一个很好的解释。而要研究谁在接触儿童，谁在影响儿童，那么这些人需要什么样的儿童语言学知识？儿童语言学怎么样支撑这一类人的工作，甚至变成他们职业上的常识，甚至制定他们的职业标准，从而让我们的孩子能够更好的成长，能够更好的发展啊！这是一个研究思路。这个研究思路与乔姆斯基的思路，我觉得是相辅相成的。但是我这是一个更现实的思路，是真正为了孩子发展的思路。中国的儿童语言学啊，在这方面应该有自己的新的创造，有自己的新的发展。时间差不多了啊，我最后是一个结束语，结束语就是结束啊。那么我今天主要是希望能够树立语言系统、百科系统、脑神系统这个灵肉3系统一体互动的语言观。语言学家不能仅仅研究语言，仅仅研究语言也是研究不好的。语言教育家不能仅仅教育语言，仅仅教育语言也是教授不好的。那么语言发展不仅受制于先天的生物性，也受制于后天的环境。语言学习是具身性的，语用能力的发展更是具身性的，是灵肉3系统的一体互动。我们要重视语言输入、语言环境的作用，研究所有12人群对儿童语言发展的影响，研究所有12人群所需要的儿童语言学知识，改善儿童的语言输入与语言环境，真正为儿童的语言发展创造条件。这就是为儿童的儿童语言学研究、儿童语言研究的视野、材料来源、设计人群、影响领域，在我这个理念下都将会发生重大变化，成为一个社会工程。谢谢大家啊！不对的地方大家批评，大家指教。
Many thanks for Professor Lee's fantastic speech. As we know that Professor Lee proposed the flash soul synergy theory, which is a very good guidance for the future developmental linguistic study for children. We hope that many researchers whose interest in child language acquisition will benefit a lot in these lectures. Now I will pass the mic to the second keynote speaker, Professor Fan Lin. Professor Fan is from the Beijing International Study University, who is chair of the second keynote speakers. Now, please, Professor Fan.
Okay, hi.
Can you hear me?
Hi, Professor Yin. I can hear you. Yeah, hi, Professor Yin.
Now it's your time.
Yeah, thank you for inviting me to participate in this symposium. Yeah, good morning, ladies and gentlemen, dear colleagues. It's my great pleasure to participate in the 3rd International Symposium of Language Science in the Era of Artificial Intelligence, Language Acquisition, Processing, and Disorders. I'm Fan Lin from Beijing Foreign Studies University, China. And it is my great pleasure to introduce the second keynote speaker for today's plenary address, Professor Jeffrey Gruen from Yale University. Professor Gruen is a full professor is a full professor at the Department of Pediatrics and Genetics and the director of the Yale Program for Learning Disability Research at Yale University, United States of America. Professor Gruhn's major research focus has been primarily in the mapping and the discovery of genes of human diseases. In 2001, he shifted his focus to mapping and identifying genes for reading disability, including DCDC2. Actually, his discovery of DCDC2 was cited as the fifth most important discovery of the year 2005 by the journal Science. Yeah, he has published over 80 peer-reviewed articles in highly regarded scientific journals, including Proceedings of the National Academy of Sciences, Nature Neuroscience, and American Journal of Human Genetics. The topic of Professor Gruhn's lecture for today is high throughput genetic screening for early detection of risk for reading disability. Now let's welcome Professor Gruhn to deliver his speech with warm applause. Okay.
Thank you.
Yeah.
Can you hear me okay?
Yeah, I can hear you, Dr. Gruhn. Yeah.
Thank you. That was absolutely lovely introduction. Thank you so much. Not so sure I'm deserved of the high praise, but I greatly appreciate it. And thank you to the organizers of the symposium today, and particularly Professor Jean Shui, who's invited me today. I think this is absolutely wonderful. And so I'm very appreciative of this. Can folks see my slides okay?
Yeah, it's okay.
Okay.
Okay,
So,
yeah, we can hear you clearly. Yeah.
Okay, that's great. All right, so I'll get started. And what I'd like to say is that, yes, it's true. We, at the Yale Program for Learning Disabilities Research, we look at the genetics of both reading and language. And we started recently this program, the Yale Program for Learning Disabilities Research. What I'd like to do this morning is go over some of the highlights of our high throughput genetic screening program for early detection for the risk of reading disability. But before I start, let me disclose a conflict of interest. Yale University has been awarded United States and foreign patents and has filed provisional patents regarding some of the content of today's presentation. So I'd like to cover 4 areas this morning. The first one is why is early identification of learning disabilities important? The second one is what is the scope of the problem? The third I'm going to try and cover is how will early identification disrupt the current educational paradigm? And where can we as scientists, where can we and educationists, where can we make a difference? And so I'd like to tell you is that all our research and development projects start in the classroom, because in fact, worldwide, 15% of students struggle with learning disabilities. And these include dyslexia or reading disability. I'm going to use these terms interchangeably today, so if I use one or the other, I mean the same thing. Specific language impairment, this is just typical delayed onset of speech in typically developing children. Verbal trait disorder, a problem of articulation. Dyscalculia, this is a math disability. And attention disorders, attention deficit hyperactivity, attention deficit disorders. But all the children I'm going to talk to you about today are in regular classes, at regular schools, and with normal or even above normal intelligence. And we test this by intelligence quotients. For today, I'm going to focus on reading. And I'd like to tell you 3 critical things to know about reading. And the first one is that dyslexia or reading disability is common. How common? Well, it depends how you set the cutoff. So if I were to do any reading performance assessment here in New Haven, let's say 4,000 children in the 4th grade, and it doesn't matter which reading performance assessment I would do, single word reading, spelling, or comprehension, and we would graph low scores down here and high scores here up to the right, and we would graph each child by virtue of their performance. Not surprisingly, if we had enough children, we would have a normal distribution with a mean. And notice it's a unimodal distribution. And reading disability is just basically where we set the cutoff. And so you'll find frequency for reading disability or dyslexia that range from, say, 5% up to 20%, because here in America, it's each individual school system will define what their cutoffs are. Not surprisingly, the higher the cutoff, the greater the amount of resources that have to be devoted to both intervention and remediation. So some tend to be more conservative, some less conservative, but an average is somewhere between 10 and 20%. So I'll check the 10th percentile here. But notice it's just the tail end, and that's how we define reading disability. This would correspond to about 5 to 10 million US school children. And let's remember that no one outgrows dyslexia. You have it for the entire life. You just may find ways around it and other compensatory tools as you grow older. So dyslexia is actually quite common. The second thing to know about reading is that all reading performance, including dyslexia, is genetic. So again, if we look at our normal distribution, we have a mean and we have a variance around the mean. And if we ask the question, to what extent do genetic factors, environmental factors account for the variance around the mean, what we see is something very surprising. Non-genetic factors account for a small part, that's environment. Genetic factors actually account for the great amount of the variance around the mean, or total performance. And now I'm talking about all of this, not just children with reading disability, but this affects how we all read to a great extent. They're highly influenced by the factors that we inherit from our parents. So what we say is that genetic factors account for up to 80% of all reading performance. It's about 60 to 80% in most studies for all of us. And what we call this is the heritability, the extent to which genetic factors can account for reading performance or the variance around the mean. So since genetic factors are mostly responsible for dyslexia and our reading performance, the mission of our lab is to discover genes associated with dyslexia, because we can identify children at highest risk at the age when intervention works best. So we developed a genetic screening panel using 2 of our studies. The first one was called the GRAD study, which is a cross-sectional study from across the United States of African-American, Hispanic-American children, ages 9 to 14. And the New Haven Lexinome project, which is our latest project. It's longitudinal and is looking at children in grades K through 5th grade. And these have been informative for children of European American ancestry, Hispanic American ancestry, and African American ancestry. And as Professor Lee already said, we've been publishing stuff pretty regularly in peer-reviewed literature. So of the 3 things that are important to know about reading, it's common, that dyslexia is common, reading performance, including dyslexia, is genetic. The third thing to know is that it can be effectively treated. So this is data from my colleague Maureen Lovett at the University of Toronto. And Maureen has been doing intervention for children with reading disability for probably close to 30 years, many of them in randomized trials. And so I'd summarize her data like this. What Maureen has done is identify children who struggle with reading in grades 1, 2, 3, 4, all the way through grade 12. And for children that, when she found them struggling with reading, she put them in one of her really extended intensive programs that last somewhere between 75 and 110 hours per year. And then she completed their intervention, and then she found those children again and retested them 2 years after completing that intervention. And here what you can see is that the children that were identified at their earliest years responded best to intervention. So early identification seems really important, because when we look at these kids that were identified in grades 10, 11, 12, in the United States, this would be high school, you can see that only about 1/3 of these kids respond. So 3/4 of children here versus roughly 1/3 here. So as we say, early identification is really important. But the key to early intervention is early identification. And the window of opportunity for that is quite narrow. So here, this is what we use in the United States, which is really prevalent in most schools, and that what we call the wait to fail model. And I'll show you what that means. If we were to chart comprehension in children pre-K through grade 5, what we'd see is mostly, but not a perfectly linear growth in comprehension over these years, from the early years of reading all the way through 5th grade. But for kids who struggle, what you can see here is they tend, as soon as the curriculum gets challenging, they fall off the curve. And predominantly in the United States, what we do is we watch them. So we watch them fail. And then they get an intervention out here, late grade 4, early grade 5. But notice that they never catch up to their colleagues in to their classmates. There's always a wide gap in these children. The alternative would be if we identified these kids early, got them into an early intervention, got them up to reading to close or at grade level. And you can see here is that the gap really narrows here. So they really do catch up. And by Maureen's data, that catch up is durable. So instead of, but the key here obviously is identifying back here early intervention. Currently, what people use are paper and pencil approaches or computer-based approaches to identify these, but truthfully, they're not very accurate, nor are they they're not sensitive, nor are they very specific down here. So they're not very good. So our approach, of course, is to use genetic screening. But how do we do this? How do we link performance in genetics and risk for dyslexia with their actual genes? And I thought I'd illustrate for you that for you this morning so you can see how that magic is done. And I'd like to illustrate one of our latest projects, the New Haven Lexinome Project, and show you within real data, which we've recently published that you can see. The goal of the New Haven Lexinome Project is to explore the extent in which genetic variants correspond with reading performance through whole genome sequence, serial behavior testing, and serial functional MRI studies. It's a longitudinal study, and these are normally developing children, as I mentioned, in regular schools, grade 1s through grade 5, that we identified in 32 New Haven public schools. We're talking about 500 children for this study for the data I'm about to show you. And we gave the parents an extensive questionnaire looking at past medical history, socioeconomic status, learning disability history, and those sorts of things. And for each child, all 500, we did whole genome sequencing. That's quite deep. It's similar to what you would do for medical testing. And in addition, we did standard psychometric batteries, which I'll show you in a moment, to assess reading, language, executive function, roughly every 6 months while they were in school. So here's an example of one of our phenotypes that we looked at in some of the assessments that we did, and they're all derived from mainstream psychometric test batteries. So these are the subtests that we use. So for example, we, in all our children, all 500, we did sight word efficiency. This is the subtest from the Tower. Most people will know what the Tower is. This is the second edition of the Tower. Sight word efficiency assesses the ability of children to read real words and their accuracy, and it's a time test. So I think they get 45 or 50 seconds, and the number of correct words that they read within that period of time is how they're scored. But some kids, as you know, will memorize words. And so what we also do is we do the phonemic decoding efficiency assessment, which is also time, but we give them non-words, words that they'd have to decode or sound out, like zoop and those sorts of things. These are not real words. They could not have memorized them. We also do 2 subtests from the Woodcock-Johnson, the letter word ID, again, to read real words, scored for accuracy, the word attack, which again requires them to decode, score that for accuracy. And then their ability to do, to read passages. So reading fluency, it's a time test, we look for accuracy, and also quiz them on comprehension. So we use these in a composite. So we put these 5 scores together in a composite score. And so we have really what I would call as a continuous score, say 1 to 100. And then we regress that information onto each child's single nucleotide polymorphisms, which are distributed through the entire genome. And I think most people are now familiar with single nucleotide polymorphisms, or what we abbreviate as SNPs. But I thought I'd just go through it one more time so folks can remember what it is. And SNPs are just that. They're single nucleotide polymorphisms. These are variations at a very specific location in the genome. The genome is made up of 3 billion bases. So in this case, we're looking at SNP with a challenging name of RS6935076. This is a real SNP. You could actually look it up. Its location is precise. It's on chromosome 8. Not only just on chromosome 8, it's on the 24,644,094th base of chromosome 8, and that's where it is. And you might say, well, how could you really tell, since the genome is only made up of A, T, Cs, and Gs? But in fact, it's defined precisely by the flanking bases on either side. And so this pattern of 13 bases or 14 bases here is present once and once only in the entire genome. And here I'm representing 3 different genotypes from person 1, 2, and 3, or 3 different children. This person has a C that he inherited from his mother, a C that he inherited from his father. So therefore, his genotype for this particular SNP is CC. You would say that he's homozygous for the C allele. And then person number 3 received a T from his mother, or his mother, a T from his mother. So at this precise location of RS6935076, we would say this person is homozygous for the T allele. And this other person, person number 2, actually got a C from one parent and T from the other. So we'd say this is a heterozygous genotype for this particular SNP. So now we know precisely exactly what base is present in each of these 3 individuals. So we can use that information, right? We can take person number CC, and we can identify a performance decoding composite score for that person, as well as the person who's CT and the person who's TT. And in fact, what we can see here is that there's a linear relationship. The more T you have, it seems the better you are on your composite score. And in fact, you don't only have to do it just for 3 children. You could do it for 500 children. You could do it for 5,000 children. You could do it for 50,000 children if you had the phenotype scores, if you had the decoding composite. And when you get to the 500 children, what you could derive is an R squared. And so with the R squared, then you can assign it a p-value. So you get a correlation coefficient and a p-value that corresponds precisely to this SNP. And since, as I said, we know precisely where this SNP is in the genome, here on chromosome 8, and then we can assign it a p-value, just like that. Here it is, that SNP on chromosome 8 with its assigned p-value. But in fact, we don't have to do just a single SNP on chromosome 8. There are thousands of SNPs on chromosome 8. So we can actually fill it in very, very nicely. In fact, we could saturate chromosome 8. And this is just one of 22 autosomes, the non-sex chromosomes. So in fact, we can do all the autosomes, beginning with chromosome 1 here on the left, all the way to the smallest autosome, which is chromosome number 22 here on the right. And in fact, the black, gray is just to differentiate the different chromosomes. Notice that the longest chromosome that we have is chromosome 1, and it's the widest because it has the most number of SNPs. And chromosome 22 is the smallest, and so it's the most narrow because it has the fewest number of SNPs. But what I was talking about is this is real data. This is the decoding composite SNP. And in fact, since we've done this with all these SNPs, roughly 1 million, that are genome-wide, we call this a genome-wide association study. And notice here that it actually looks like we're getting a signal. In fact, there is a signal coming out of chromosome 19 right here. And that signal lands on a gene we had never heard of before called GARRE, G-A-R-R-E, GARRE1. So we went back to test how good GARRE1 is as a gene associated with reading disability. And we retested all our 500 kids looking for whether they have the deleterious allele, the minor allele at that particular SNP, or they are either carrier 1 or 2 of those versus not being a carrier at all. So the children here, for each of these assessments, and we have letter word ID here in the top left-hand corner, word attack here in the bottom left, and comprehension, or in orange, are at least carriers of 1 or 2 copies of the deleterious allele of GARRE1, and blue is they carry no copies. And what you can see here is as we zoom in on this, is we have data from the start of grade 1 in the Lexinome project to the end of grade 1. to the start of grade 2, the end of grade 2, the start of grade 3, the end of grade 3, all the way to the start of grade 5. And in each instance, the children that carry one copy of the deleterious allele of GARI1, the minor allele, are always performing less well than their counterparts in the same class in the same schools. Interestingly, we had not known of GARI1 before, but when we look on the publicly accessible databases like GTEx, this is a gene whose expression, whose protein seems to be really important to brain development. It's expressed highly in brain, in particular in the cerebellum of the brain. This is really, really interesting because there are cerebellar cortical loops that we know of that are well documented both structurally and functionally. And it plays into the cerebellar theory of dyslexia, which was first put forward by Nicholson and Fawcett in the 1990s, in which they said that dyslexia is associated with deficits in cerebellar-related functions like dystonia, time estimation, skill automatization, and balance. And so it would appear, as Nicholson and Fawcett said, is that cortical cerebellar circuit has an important role in reading. And indeed, this would support that theory. So when we look at our current approach, a wait to fail model, and now we have a method to identify these kids by putting these SNPs together into a single panel. So our question is how to disrupt the current educational paradigm. So as Professor Lee mentioned, we've been publishing here, doing this stuff for almost 25 years, but yet all our information seems to be retained in the academic literature. Are we really helping children? How do we get this information into the classroom? Are we really disrupting the current educational paradigm? And I think that's a really questionable. So in our new program, the Yale Program for Learning Disability Research, we've really began to address policy changes here in the United States. And let me tell you what I'm talking about. Here in the States, in the last 5 years, dyslexia and learning disability policies at the state and the federal government have improved significantly, most notably in the areas of teacher training and universal dyslexia screening in grades K through 3. It's mandated, but it's not funded. Funded comes only through local levels. In fact, if you look at statewise, state by state here in green, of states that have actually implemented universal evidence-based screening, it's not all 50 states of the United States. Federal clarification that dyslexia is a permissible term can be used on individual educational programs. And 32 states have provided a list of approved screeners, but none of them include genetic screening. They're all conventional paper and pencil and computer-based screening. So the goal at our new program is to pass legislation that includes reliable and validated genetic screening for risk of dyslexia and get it on the approved list of evidence-based screeners. Once implemented, this would allow parents to screen their child as early as birth for the risk of dyslexia. It would require school districts to take these results into account when they create their IEPs, the individual education plans, and the 504 plans. And it would enable children to receive an evidence-based reading instruction or intervention method at the various earliest stages of reading acquisition when it is most useful. So these are the take-home messages for this morning. I presented a case, and I hope it came through, is that early identification of children at risk for reading disability is the key for effective intervention. That the current wait to fail model that we predominantly use in the United States for identifying children with reading disability misses the window for deploying effective intervention. In other words, most kids are missed. And so even if you identify a child in later middle school, or early high school, even late high school, and even if you were to give them an effective intervention, it's not that useful. Only about 20 to 30% of those kids will respond. The key is to get it early and get it in that narrow window when it is highly effective. Traditional paper and pencil testing or computer testing are unreliable for identifying children with reading disability at an age when intervention is most effective. So if intervention is really, really useful in grades 1 through 3, but the testing is not particularly accurate or specific at those times. So there's a mismatch between testing and intervention so that we have a basic inability to identify these kids and to deploy a really good intervention program. And finally, our high heritability, the 60 to 80% of the variance that we describe around the mean for reading performance, and what we mean is mostly comprehension, makes genetic screening the optimal method for early identification of children at risk for reading disability. So I think at that point, I'd like to stop and thank all the wonderful people that it takes to do these sorts of studies, all my wonderful, fantastic collaborators, and the generous funding of private donors, as well as the National Institutes of Health, and support, of course, from the Yale School of Medicine. And I'm happy to take any questions if people have any.
Okay. Professor Gruen, thank you for your insightful and interesting speech. We're delighted to have this opportunity to learn about your innovative work. Professor Gruen's speech is about genetic screening for early detection of risk for reading disability, which is informative and illuminating, broadening and widening our horizon. Yeah. As we know, that children with a diagnosis of dyslexia usually suffer from specific difficulties with reading and spelling words accurately and fluently, posing significant challenges to their academic attainment. They are at risk of lower life chances if they do not receive the support they need, in particular early intervention they need. Therefore, it is important to identify children at risk before they suffer from academic failure and the subsequent consequences, including lower self-esteem or poor self-image. In his talk, Professor Gruen presents in detail the why, what, and where of genetic screening for early detection of risk for dyslexia. I'm sure all of us have learned a lot from Professor Gruen's insightful speech. I'm sorry, due to our tight schedule, I'm afraid we do not have time for questions and further discussion. I'm sorry to say that this session will have to stop here. If you have any questions, you may contact Professor Gruen via email and discuss with Professor Gruen. We would move on to the next section. Next speaker. Once again, I would like to express my sincere appreciation to Dr. Gruen, Dr. Gruen, on behalf of all the audience. Thank you. Okay, thank you, Professor Gruen. Okay, the next, today's the third. Hi, Professor Francis. Now Professor Norbert Francis, the third speaker, keynote speaker for today's plenary address. I'm very pleased to introduce Professor Francis, who is a professor and supervisor, doctoral supervisor at Northern Arizona University. Professor Francis is a very prolific scholar. He has published more than 150 publications. He has more than 150 publications in highly regarded journals. And his research interest include, but not limited to bilingual studies from cross-linguistic and cross-cultural perspectives, the role of language awareness in literacy and second language learning, awareness of form and pattern in literacy assessment, the use of first language and second language in the classroom. Professor Francis' topic for today is domain-specific and domain-general aspects of language development, research on learning processes related to System 1 and System 2. Now, let's welcome Professor Francis to deliver his speech with our warm applause. Okay. Yeah, Francis,
Hello.
Professor.
Good morning, everyone.
Yeah.
Let's see if I have my screen. Can everyone see my screen?
Yeah. Yes. Yes, Professor.
Let's see if I can get it right in
Yeah.
the middle there. Okay, very good. Well, thank you very much, first of all, for the very kind invitation from the organizers of this conference to let us share with you our recent findings from our work. If we run out of time for questions, please write to me at my email, and I can send you the references for this presentation and all the slides, so you can look at them at your leisure. So the topic is on domain-specific and domain-general aspects of language development. And there's two words I wanted to emphasize in the title, and that's aspects and processes. And they're both plural. And the main idea of the whole talk, if I would say it in 1 minute, is that language development and language in general are internally diverse. In other words, there are many aspects of language development, not just one single undifferentiated aspect. And there are many learning processes, not just one learning process. So we could think of these as perhaps components of language development and language use and language competence. And that'll be the main idea throughout. And when we look, I think looking at the internal diversity of language development, and this applies to all cognitive abilities, I think that will help us in understanding better how to design research methods and research approaches, especially for developing children. And in particular, I want to emphasize in particular in the case of developing children who are learning 2 languages, learning one language first and maybe learning a second one later in life. So before I start, I just want to make one point about what's called a common origin. The common origin of language is one. It is one. We all share a common origin, and you can see where that is. That means, at least for us in our project, and maybe it's controversial, but if there's a discovery about language development, and it's a true discovery that we could actually confirm, no matter where that discovery occurs, over here, in the Americas, in East Asia, it should apply in some way or at some level to all other language communities, and that would be without exception. And so that's the idea here. And the other aspect of this map is that in our project, we've been promoting an interaction amongst researchers that we think will be very productive in the years to come, and that is between these 2 parts of the world. By pure coincidence of the migration of our species, this was the last division of the many divisions in the migration of humans, and these 15,000 to 35,000 years ago, that's the most recent division. And so it's maybe a coincidence that coincides with many aspects of the languages of the Americas over here, and the languages of East Asia. have in common. There are many aspects in particular, of bilingualism and multilingualism that are shared between these two parts of the world. In fact, more so than other parts of the world. I just wanted to mention that. And because of that, we want to, with our project, our small little project, to encourage and to promote this kind of a dialogue across the Pacific Rim. So let's see if I can, there we go. Yeah. So the place to begin when we think about the components of languages is to start with language abilities. Because language abilities is where we can observe and actually measure what might be underlying in the abilities. So language abilities, or language ability in particular, I think is a good place to start. For the implementation, language ability for its implementation requires the participation of these underlying competencies. Some of them will be system 1 and some of them will be system 2. And in the next slide, we'll define what these are. For example, in reading ability, we just listened to a presentation on reading ability. Well, reading ability is a complex ability, and there must be underlying competencies of great diversity. Just think about very quickly, there must be an interface or an interaction between the visual system and the language system that is implemented in a very close interaction, in a very close interface in reading. In narrative ability, think about it. It's not just language, it's not just linguistic ability, but it's the ability, for instance, to tell a story in a logical and chronological order. And so these are different competencies that come together in performance in abilities. So that's why we say it's a good idea to start with at the ability level. So let's very quickly define our terms here. You can look them up on the internet, System 1 and System 2. And very often other researchers will use, instead of System 1 and System 2, they'll use the words Type 1 and Type 2 to avoid the idea of a system, an enclosed system, because it's not really an enclosed system. It's a word that's in psychology that has been used a lot by psychologists, and we have borrowed it from psychology to apply it to language abilities. So language abilities, we would say, typically need the combined activation of different component competencies and processing mechanisms. And System 1, or Type 1, would be the kind where kinds of competencies and mechanisms that are evolutionarily primary. They're very early in our evolution as humans. Whereas with System 2, they are secondary. They came later. Literacy would be an example, okay? Literacy is a very recent ability that humans have begun to learn. So this literacy will be predominantly controlled by Type 2 or System 2 kinds of competencies. A System 1 competency will have early emergence, okay? Again, and System 2 will be late. The next point, I think, is the most important here. It's the one that describes the two types. System 1 is specific. It's a domain-specific competency. It's specific to something very specialized. Whereas with System 2, these will be what we call domain general. They will be of the more general type. Domain general, an example of that would be in working memory, for instance, how we can control attention and inhibition of information that we have to process. So that would be an example of a general capability versus a specific capability. I think I was lucky to follow Professor Gruen because he gave a good example of a domain-specific competency, which is phonological competency, and in the case of SLI, specific linguistic impairment, that's an impairment of another System 1 competency that's very specific, in this case, to grammar. So it's when the grammar is impaired. It's a rare syndrome, relatively rare, but it's a good example, I think, of domain specific. And I'm also lucky to come before Professor Kebab because she will talk to you about the lexical quality hypothesis. This is an old hypothesis, been developed for quite some time, and I think it has been promoted beyond a level of hypothesis. It's a very important concept. So pay attention to this concept. And this one, in this case, will correspond, at least in our model, to type 2 or system 2. So you get 2 examples already. System 1 here is automatic acquisition versus deliberate learning in system 2. The performance is also automatic versus a system 2 process would be more demanding cognitively, that requires more attention on our part to implement. System 1 is implicit. That means it's below awareness. And system 2 would tend to be subject to awareness, and especially of metacognition. We can talk about metacognition later. In system 1 or type 1, within a population of normally developing children, these are universally acquired. And the developmental trajectory is an early plateau, right? System 2, we expect, if it's really truly type 2, that there will be significant variation and that it's open-ended in its development. So here's some examples. I think that the 2 best examples that we'll return to during the presentation is language 1 and language 2, L1 and L2. I'll just use L1 and L2 now. And I think you're familiar with this dichotomy or this distinction here. For your first language, it must be during childhood. It must be during a critical period. It is spontaneous acquisition, does not require teaching. Specific language impairment aside, there's an early developmental plateau, okay? In contrast, L2 is second language learning. It comes typically later. It can be learned during early childhood, but later and even by adults, normally. Whereas if you miss the critical period in your L1, that typically results in impairment to your ability to use language. That's why it's critical. That's why they call it critical period. In language 2, focused learning and metacognition do contribute to the rate of progress and ultimate attainment, okay? But that's not true for L1, okay? We know that. This is one of the robust findings of anthropology of first language acquisition across cultures, that children will acquire their first language whether their parents try to teach it to them or not. It requires exposure and engagement with the language, and that's all. We call that sometimes simple immersion. With simple immersion, you will acquire the core grammar of your mother tongue. That's what we say, uniformal ultimate attainment. When we refer to the ultimate attainment, we're referring to the core grammar of your mother tongue. But in second language, we know this also as a robust finding, that there is wide variation, even after years and years and years of of quality exposure to your second language, there will be wide variation from one individual to another. Some individuals will be virtually or near native, almost indistinguishable from L1 speakers, and others will not be, even after many years of quality exposure and even practice and learning to the second language. We know this to be a fact. So I think L1 and L2, I think, are the best examples of System 1 and System 2. So we'll go back to these later. System 1 looks like this. This is the curve for System 1. It's an early plateau. If we gave a test of morphology and syntax of the core grammar, you would see that amongst normally developing children, there is a early plateau. It probably even comes earlier than I'm showing here. I'm showing it at second grade. It comes earlier than second grade even. But for instance, let's take the example of reading achievement. This does not have an early plateau. There is wide variation from one child to another, as we saw as we heard from Dr. Gruen, and it is open-ended. At the end of 8th grade, I just put 8th grade there just arbitrarily. It's not over there either. Open-ended means it continues to develop even after 8th grade. And so that's what we mean by open-ended and significant variation from one child to another. So you might say, well, maybe I'm trying to make this too complicated. System 1 competencies, maybe they're just easier to acquire. But that's not a good way to think about it. Here is a System 1 competency, grammar, okay? And every language has these kinds of problems in syntax. And we won't go into detail, we'll skip over this carefully, but the native speaking child at age 5 or 6 knows the difference between these two constituents of syntax and knows unconsciously that they had to be treated differently in the grammar. He has never been taught this by their parents. He couldn't have been taught this by their parents because their parents don't know this, okay? And even amongst linguists, these theories are controversial, but nevertheless, the normally developing 6-year-old child with simple immersion, simple exposure to the language, will develop very sophisticated algorithms for processing the syntax of the first language. And this is System 1. It does not require any instruction at all. So here's our first project that we want to talk about. We have one project in East Asia. It's led by my colleague, Professor Le Nam from National Tsinghua University in Taipei. And here's one of the problems that we think about, okay? We think about, it's about the learning of Chinese as a second language, okay? That's the main idea here. And so we're thinking of L2 learners of Mandarin who are L1 speakers of different languages. And this is just a partial list. And so how would the experience of second language learning be different for these different languages? And you can start with one of the dialects of Mandarin itself. There's a very nice article I put in references on this case of Yunnan Mandarin, and you can see it when you get the references. Then there's from within Sino-Tibetan, the same family of the Chinese languages. We're talking about now a very close language, Cantonese. Within Sino-Tibetan, but not a Chinese language. Then outside of Sino-Tibetan, but a language here, Vietnamese, with a long history of contact with Chinese, okay? Some people, Vietnamese has been affected so extensively by its contact with Chinese that some people think of it even as part of the same family, which it is not, okay? Very interesting case. Uyghur would be from outside of the Sino-Tibetan family, but with less influence than Vietnamese, much less. and a completely foreign language, a completely foreign language like English. So how would these L2 experience, the second language experience of these different individuals be when they learn Chinese? Okay, and of course in reverse, we could look at the process for L1 speakers of Putonghua who are L2 learners of these other ones. And what we're saying is System 1 processes and System 2 processes permeate all of these examples. They don't explain all of them. There's other factors. We're not saying these are the only factors. There's other factors like motivation, for instance, which is very important. But System 1 processes, which are, remember, these are the automatic, and System 2, which are the deliberate learning, permeate all the experiences of second language learners of Chinese and of any other language that is an L2 language. We're making that claim. Okay, just to show you, just to illustrate what I just said about motivation, for instance, learning English is very popular amongst speakers of Chinese. We know that, and we know also that speakers of Chinese as L1 are very successful in learning English, even though we put it down furthest away from Mandarin. But the obvious explanation is that there are other factors involved than just System 1 and System 2 processes. Okay, I just wanted to say that. So our project focuses on a population of immigrants, in this case, from Vietnam to the Taipei area. And they have different circumstances, like you saw on the previous slide. And the first group we had, these were, in this case, they were all young women, and they were this group was a monolingual Vietnamese speakers, okay? And in a study of self-report, we interviewed them 20 years after their arrival to a Chinese-speaking society, and about their progress, their early progress in learning Mandarin Chinese. And it was very interesting to listen to these accounts. I just want to say, I'll repeat again, they were self-report, and therefore it has a big limitation, but it's a good way to start a project, to get the testimony of actual learners, and then start from there. And then that will give you hypotheses about what kind of actual direct assessments of Chinese as a second language that would be the most appropriate in this case. Well, here's some of her homework that she did. We picked this one as it happens to be the most advanced learner. As you can see, she was already even when she was still in Saigon before she got her visa. They went to a learning center to start learning Chinese. The Vietnamese government is very how should we say helpful in facilitating the immigrants' adaptation to a Chinese-speaking society. So you'll notice here a typical strategy here of translating the Chinese here. But what we found was even more interesting was the use of Vietnamese pinyin. It's a new topic in the literature. In fact, I don't think we found even a single article on it, but I suggested it's a very interesting topic for further research there. And then there was a second population of Cantonese speakers. And I'm going to leave you with this one because the comparison here was with the Vietnamese monolinguals, they did very well. They did very well with learning Chinese as a second language. It was actually surprising, their report of progress in learning Mandarin Chinese. And in this case, it was largely, except for a few students who studied in school,